Imagine trying to read a book, but you don't even know the alphabet. For a long, long time, that's what our own DNA was like. A book written in a totally alien language. But then, one absolutely brilliant technique came along and gave us the key. It let us finally read the code that makes us who we are. Today, we're diving into that Nobel Prize-winning method, Sanger sequencing.
So here's the plan. We'll start with the absolute basics, the alphabet of DNA. Then we'll get into the clever trick that makes the whole thing possible. We'll see how the method grew up, from a slow, old-school process to the high-tech version we use now. And of course, we'll look at how to actually read the results. And finally, talk about why this discovery is still so important today.
All right, first things first. Before you can read a code, you gotta know the letters. So let's break down DNA into its most fundamental parts. Every single strand of DNA is built from these little units you see here, called nucleotides. Each one has three parts: a phosphate, a sugar, and a nitrogenous base. Now, it's that base that's the really important part for us. It can be one of four things: A, T, C, or G. You can think of those as the four letters in the genetic alphabet. Our entire goal here is to figure out the exact order of those letters. So, a nucleotide is the what we're trying to read, right? A single character in this massive code of life. But how do these characters link together to form the words and sentences of our genome? Okay, look really closely at this diagram. See that specific spot on the sugar molecule? It's called the 3' hydroxyl group, or just an OH group. This tiny chemical group is, well, it's everything. It acts like a hook, letting the next nucleotide latch on and make the DNA chain longer. If that hook isn't there, the chain can't grow. Remember that hook. It's the key to this whole story. And this is where the genius of Frederick Sanger really shines. He had this incredible insight. If that hook is what builds the chain, then removing the hook is how you can read it. To figure out the sequence, you just have to find a way to controllably stop the chain from getting any longer. And this is the core idea right here. It's beautifully simple. If that all-important hook, the 3' OH group, is missing, the whole process just grinds to a halt. It's a chemical dead end. And that's exactly what Sanger was looking for. This slide shows the trick perfectly. On the left, you've got a normal, everyday nucleotide with its little OH group hook all ready to connect. Now, look to the right. That's Sanger's secret weapon, a dideoxynucleotide. See that same spot on the sugar? The hook is gone. It's been replaced by just a hydrogen atom. It's such a tiny change, but it has enormous consequences. And that's the light bulb moment. Once the cell's machinery adds one of these modified nucleotides to a growing DNA chain, there's nowhere for the next one to attach. The process just stops, dead in its tracks. It is the ultimate biological stop sign. So this special molecule is called a dideoxynucleotide, or DDNTP for short. You can think of it like a molecular imposter. It looks enough like a normal nucleotide that it gets added to the chain, but because it's missing that critical hook, it's a dead end. It is, literally, a chain terminator. Okay, so we've got this super clever trick, but how do you actually use it in a lab? Well, this is where the method comes in, and it's a method that has changed a lot, from its original form to the sleek version we have today. So back in the day, the classic method involves setting up 4 different reactions. You'd have 4 tubes, and each one had all the normal stuff needed to copy DNA. But then, you'd add a tiny amount of a specific chain stopper to each tube. For example, tube 1 got the A stoppers, tube 2 got the T stoppers, and so on. What you ended up with was 4 different sets of DNA fragments, each set ending only at a specific letter. Now, this was absolutely revolutionary, don't get me wrong, but as you can probably imagine, running 4 separate reactions, using radioactivity, and then analyzing it all, it was slow and really manual. There just had to be a better way. And there was. The modern upgrade is just so much more elegant. Instead of radioactivity, each of the 4 chain stoppers gets its own unique fluorescent color tag. Green for A, blue for C, you get the idea. This means you can just toss everything into a single tube and run one reaction. Then you let a laser automatically figure out the color, and therefore the letter, at the very end of each DNA fragment. So once the reactions are finished, you've got a tube that's just full of DNA fragments, all different lengths, and each one ends with a specific known letter. How in the world do you turn that jumble into a coherent sequence? This is the classic result. It's called an autoradiogram. Basically, you separate all the DNA fragments by size on a gel. The shortest fragments zip down to the bottom the fastest. To read the sequence, you literally just start at the very bottom and work your way up. You find the lowest band, see which lane it's in. Let's say it's the A lane. And boom, that's your first letter. Then you find the next lowest band, check its lane. And you just keep building the sequence, one letter at a time.
Now, compare that to the modern output. Oh, it is so much cleaner. Instead of a gel, the fragments fly through a tiny little tube sorted by size. As each one passes a detector, a laser zaps it and reads the fluorescent color of the final base. The machine then spits out this graph, a chromogram. Each colored peak is a letter. It's automatic, it's fast, and it's super easy to read.
I mean, the leap forward here is just staggering. The process went from 4 separate lanes to 1, from handling hazardous radioactive material to using safe fluorescent dyes. And maybe most importantly, it went from a scientist having to painstakingly read a gel by eye to a computer doing it all automatically with a laser. Sanger's sequencing was a monumental achievement, there's no question about it. But it's really important to understand where it still shines today and also where it kind of falls short in this new era of genomics. This number right here, around 900, it really highlights a key limitation. A single run of Sanger sequencing is incredibly accurate, but it can only read a stretch of about 900 DNA letters. To put that in perspective, that's like reading one single page from a library that has millions and millions of books. So this slide really lays it all out. The huge strength of Sanger is its accuracy. It's so reliable that it's still called the gold standard for verifying results. It is perfect for small, targeted projects. But the limitations are pretty clear. It has low throughput. You can't do very many sequences at once. That makes it expensive and time-consuming for big jobs. And as we just saw, that read length is pretty limited.
This really puts it all into perspective. If you need to read one specific 900-letter paragraph with perfect accuracy, Sanger is absolutely the tool for the job. But trying to read the entire 3 billion-letter human genome this way, it would be impossibly slow and wildly expensive. But we cannot overstate its importance. For decades, this method was our Rosetta Stone. It's what taught us the language of genetics in the first place. It powered the original Human Genome Project. It literally laid the foundation for everything that has come after it. Sanger's method taught us how to read the book of life, one short page at a time. And it paved the way for the incredible technologies we have now, technologies that can read entire libraries of genetic information in a single day. So that leaves us with a pretty profound question. It's not about if we can read the code of life anymore. It's about what we choose to do with that knowledge.